Hello, my dear students. So this is a continuation of our previous class. So stem cell and neurodegenerative disease that we already discussed in the previous class. Okay. So neurodegenerative disease hold an opportunity for the clinical use of stem cell in the neuroscience. The discovery of this neural stem cell MSCs or subsequent research have been nullified the previous idea that the adult CNS was not capable of neurogenesis. Central nervous system was not capable of neurogenesis. So here, indeed, this neurogenesis occurs throughout the life. It will happen throughout the life. There's neural stem cells are believed to reside within the subventricular zone of the lateral ventricular wall and subgranular zone of hippocampal dental gyrus, where this neurogenesis will occur, where it will helping in curing such type of neurodegenerative disease. Okay. So this neuronal stem cells which will give rise to a glycal restricted precursor and neuron restricted precursor, both of which differentiate into astrocytes, oligodendrocytes, or neurons. So as we know that the stem cells have the ability to convert it into almost all type of cells. So the neuronal cells can be synthesized or derived or differentiated from this neural stem cell. Another studies demonstrated that transplanted NSCs isolated from a 9-week-old human fetus has the ability to differentiate it into neural cells, and also it will improve the cognition in aged adults. Okay, so these are all related to the neurodegenerative disease. Hence, this idea of using the neuronal stem cells for neurodegradation treatment are efficient to be happen. Then however, this neuronal stem cells are not so easy to access as a source of stem cell for possible use for the treatment of neurodegenerative disease. Previously, many studies have been suggested that use of bone marrow derived mesenchymal cells, which is involved in the regeneration of neural cells. Since this mesenchymal stem cells are present in the bone marrow and are relatively easily accessible within the human body. Okay, so now there are different diseases which is related to this neuronal disease. The first one is Parkinson disease. Okay, how the stem cell therapy which is involved in curing the Parkinson disease, which was first described in 1817. The PD, which is mentioned as Parkinson disease. The second most common neurodegenerative disease. The first is an Alzheimer. First is an Alzheimer. The second most common neurodegenerative disease is the Parkinson disease. The neurological degenerative disease that will result in the loss of dopaminergic neurons within the substantial nigra, leading to the loss of motor function. Okay, the dopamine and dopaminergic neurons will be lost during this Parkinson disease, will be affected. So this Parkinson disease itself is not fatal, but complications from the disease which may lead to death. Current treatment for this Parkinson disease include drugs and other surgery may be possible, but this treatment are purely palliative. Okay. So that's what it is ineffective. And the current drug treatment for this Parkinson disease supplies the surviving dopamine neuron with L-dopa, which is converted into dopamine. So for the particular Parkinson disease, people will give a drug called as L-dopa, which is converted into dopamine, so that it can able to transport or signaling the okay. So eventually, however, all of the remaining dopamine neurons die, and the treatment with the L-dopa is inefficient. Okay, there is a drawback for this L-dopa treatment because sometimes it leads to craziness and they may affect the mental. It will create some mental problems continuously if you are taking them drugs. Okay. So one of the alternate is this MSCs, mesenchymal stem cells, has been proposed a potential treatment for Parkinson disease. Park and colleagues, which they investigated the use of mesenchymal stem cells in this Parkinson disease mouse model to observe a potential neuroprotective effect on neuronal growth. And the mesenchymal stem cells significantly preserve the number of dopaminergic neurons and tyrosine hydrolyzed positive cells in vitro and in. Okay. So in a different study proposed by Mural and colleagues, they found out that adult olfactory stem cells for the recovery of dopaminergic neurons in Parkinson's disease. The adult olfactory stem cells were differentiated into NPCs and can be able to or capable of becoming a dopaminergic-like neurons both in vivo and in vitro. So studies were supporting that with the help of stem cell therapy, either NPCs or MSCs can be used as an alternative for L-DOPA and can efficiently cure this Parkinson's disease. Then this neuronal stem cells has also been investigated for the use of possible treatment for Parkinson's disease. Certain Shugara and colleagues, they tested that analysis on the behavioral benefits and protective effects in Parkinson's disease. Then when we are using this analysis, we are immediately transplanted after 6-hydroxydopamine relation formation in mice. Tyrosine hydrolyzed neuron were protected and of course PD symptoms were reduced. Okay, while researching that, they found out neuronal stem cells can able to reduce the symptoms of Parkinson's disease. That's what the ultimate report is generated. Okay, here you can see that it's a diagrammatic representation. It's just saying that the culturing of the cells. Okay, the culturing of the cells. Starting from, they will take the media and they will culture clonal expression in the growth medium, and after the second step, which is involved the replaced cells on the surface to promote the aggregation. Aggregation is happen and growth, which will be taking for the 4 days, which is happening here. Then the next stage, it is mentioned, is replicating into the original growth medium for 24 hours. We'll take the cells and they'll grow it and can be replicated within 24 hours. Then should be replaced. The next step, step 3 here, they'll replace the growth medium with ITSFN, and only those cells can expressing the nestin will survive, will grow for 10 days. Okay, it's a marker to identify the neuronal cells. Okay, they'll know this is where they will identify with the help of ITSFN. Okay, so nestin producing expressing cells can able to survive here, other cells will die. So we take for growth for 10 days. After that, here this is a step 4. They'll replace the nestin positive cells and they will add BFGF. Okay, BFGF, that is fibroblast growth factor. Okay, basic fibroblast growth factor. Then lamins, HSS, and the FGF, the fibroblast growth factor 8, also be used here. They will replace. Okay, to for the selection purposes we have. Okay, so here you can take it and then removing the basic fibroblast growth factor from the growth medium and callus which will causing the cells to stop dividing growth for 16 days to create a dopamine neuron. Here you can get the dopamine producing neurons if you are removing the basic fibroblast growth factor from this step, and you can get here. Then this step, this arrow, it is indicating the next process state. Maybe if you are removing that FGF8, that is fibroblast growth factor 8, so that you can the growth medium, from the growth medium, if you are removing this FGF8 instead of this BFGF to create a serotonin neuron. Serotonin neurons can be able to produce. Dopamine neurons can also be produced if you are removing the BFGF, that is basic fibroblast growth factor. If you are removing that, you will get a dopamine neuron. If you are removing the fibroblast growth factor 8, you will get serotonin. That should be going for a conversion. Okay, so this is where the culturing the neuronal cells for neurodegenerative disease. Okay, next is about Alzheimer's disease. Okay, the second is Alzheimer's disease. Okay, I already mentioned that Alzheimer's disease is a condition that will affect the brain. The symptoms are mild at the first and became more severe over the time. It is caused, it is named after Alzheimer, who first described this condition in 1960. Okay, a common symptom of this Alzheimer's disease include memory loss, language problem, and other unpredictable behavior. You may see that Alzheimer's is a continuous memory loss. The people, they don't know who are there and how they are behaving, and that's some mental problem. One of the main feature of the condition is the presence of plaques and tangles in the brain. There is a formation of plaques and tangles in the brain. Another feature is the loss of connection between the nerve cells or the neuron that leads to Alzheimer's disease. These features mean that information cannot pass easily between the different areas of the brain or between the brains of the muscle organs. Due to the lack of information passing from one area to another area, it leads to craziness. As a symptom worsened, it became harder for the people to remember reason, even to the reason, and to recognize people they know. Eventually, a person with Alzheimer's disease may need full-time assistance. They don't know who they are. According to the National Institute of Aging, Alzheimer's disease is the sixth leading cause of death in US. Okay, so that is about an Alzheimer's disease. Then how it is happening. Hippocampus, where memories are laid down, the frontal lobes, it is a seed for logical thought are affected. But first, it will affect the hippocampus, and there's a logical thought which were present in the frontal lobes have been affected. The affected nerves in Alzheimer's disease will produce a different neurotransmitter that is called as acetylcholine synthesized. Then apparently in Alzheimer's disease patient, apparently in the Alzheimer's disease patient, the normal repair system is just not fast enough to compensate the disease. Okay, stem cell could be unable to provide a boost needed to balance ongoing the dispersion. So the destruction of the nerve or neurons can be replaced with the help of stem cells. Okay, that's what mentioned. And here it is mentioned recent research have been found on genetic component to Alzheimer's disease. A specific allele of the apolipoprotein, that is apolipoprotein. Okay, apolipoprotein E gene predisposed a person towards Alzheimer's disease. Okay, so this is the reason. That means if the stem cell could be engineered to eliminate this allele, if you are removing the particular allele, you can able to get rid of this Alzheimer's disease. That is the aim they mentioned. Okay, so unlike this Parkinson's disease, this have been no fetal cells trial in human have been done. But research with mice is quite encouraging. In April 2001, there is a researchers in Chicago reporting the injected aged memory implanted rat in with neuronal stem cell. They replaced neural stem cells. He found that stem cells spreading throughout their brains and the existing cells seem to be regenerated. Then the rat were trained in a water maze and the injected rat performed better than the control. Okay, so once it is treated with the neural stem cells, they'll induce Alzheimer and they can able to cure the Alzheimer in the rat. In fact, some of the elderly rat actually performed better than the young rat. That was an important fact it is mentioned. The fact the elderly rat actually performed better than the younger rat without the memory impairment. Okay. So one of the interesting aspect of this research is that human embryonic stem cells were used as a source for transplant. This implies that embryonic stem cell may exhibit specific independent plasticity, finding a way to fit in even in a different animal. So if you are using such type of system, it can be fit in any type of animals. Okay, so that's what they successed in this Alzheimer's disease against the rat. Okay, so while replacing that stem cell, they can able to find the rat can able to regain their memory and it can perform well. So in 2003, as a group of researchers lead to in the school of medicine at Tokyo reported that technique to make an Alzheimer's disease cell from embryonic stem cells. Okay, they genetically engineer the embryonic stem cells and express the B64 to I mutation that leads to Alzheimer's disease. This provides an excellent model for Alzheimer's disease, highlighting the ability of the embryonic stem cells to advance experimentation as well as the therapy. They believe their protocol is flexible enough to provide model for many degenerative diseases in human as well. So finally, they come to the conclusion that with the help of embryonic stem cell and neuronal stem cell, they can able to replace and they can cure the Alzheimer's disease. So if it is used for Alzheimer's disease, maybe they can use it for some other neurodegenerative disease too. Okay, so that's all for today. So thank you all. See you in the next class. If you have any doubts, you can contact me. I will clarify you. Okay, thank you.